Hello, myself Gautam from 316. Today my topic is relationship between the primary and the three-dimensional structure of protein. This relationship between primary and three-dimensional structure of protein can be illustrated by four main stages of protein organization. First is primary structure, second secondary structure, third one tertiary structure, and fourth one quaternary structure. First, we will discuss about primary structure. This is the linear sequence of amino acid in the polypeptide chain. In this, each amino acid is connected by peptide bonds. Specific order of this amino acid determines the protein's properties and how it folds. Like in example, there are hemoglobin, valine, histidine, leucine, threonine, proline, and glutamate. And second, we'll discuss about the secondary structure. It is also known as local folding patterns. In this, polypeptide chain begins to fold into repeating patterns like alpha helix and beta sheets. In this, structures are stabilized by S2 bonds between the backbone atoms of amino acid. Example, alpha helix and beta plated sheets. Third one is tertiary structure. In this, the full three-dimensional structure forms and interactions between side chains of amino acid occurs. In this structure, it is stabilized by the various forces including hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. And our example of tertiary structures are alpha keratin and carboxypeptidase A. Fourth one we will discuss about quaternary structure. It is also known as multiple subunit assembly. In proteins made up of more than one polypeptide chain, the quaternary structure describes how these chains are arranged in relation to one another. Like example, hemoglobin, DNA polymerase ribosomes. The three-dimensional structure of protein, often referred to as its tertiary structure. It is the overall shape that a protein that takes after folding from its linear chain, also known as primary structure. This shape is crucial because the functionality of protein is linked to its 3D structure. Here's a breakdown of the key aspects involved in the function of and properties of three-dimensional structure. First one, tertiary structure formation. It forms as a result of interactions between the amino acid chains in the protein. This bonding and interaction takes place by several steps. First one, hydrogen bond. Second, ionic bond. Third one, hydrophobic bond. Interactions. Fourth one, van der Waals forces. And fifth, disulfide bonds. In hydrogen bond, it forms between polar side chains. In ionic bonds, it occurs between opposite charge side chains. Hydrophobic interactions, non-polar side chains, tends to cluster together inside the protein. In van der Waals forces, weak attractions between closely packed atoms. And in disulfide bonds, strong covalent bonds between the sulfur atoms of cysteine residues. In the formation and importance of three-dimensional structure, there is second topic, domain and motifs. Larger proteins often have domains which are distinct functional and structural units within the protein. Each domain can fold independently. And motifs are small structural elements within the tertiary structure such as helix loop, helix motifs, or zinc fingers, which are involved in particular biological functions like binding DNA or other proteins. Third one is stability of the 3D structure. In this, 3D structure of protein is generally stable but can be disrupted by factors like temperature, pH change, or chemicals, a process known as denaturation. In this, denaturation can cause the protein to lose its functionality to its atoms. Fourth one is functional importance. In functional importance, there are three parts: active sites, protein, and binding specificity. In active sites, in these enzymes, the 3D structure creates pockets known as active sites. In protein interactions, the 3D structure allows proteins to interact specifically with the other proteins or molecules. In binding specificity, in this, the shape of proteins allows them to recognize and bind to specific molecules with high specificity, such as hormone binding. And fifth one is folding pathways. The folding of proteins into thin correct 3D shape is usually a highly regulated process. Separine proteins often assist other proteins in achieving their correct folding and preventing misfolding their atoms. And our sixth topic is quaternary structure. In some cases, multiple polypeptide chains come together to form a quaternary structure like hemoglobin, which consists of 4 subunits. And in the conclusion, we will say that the three-dimensional structure of protein is vital to its biological function and stability of the protein and its atoms. This structure unite from the folding of the polypeptide chain driven by interactions between the side chains of amino acids. The proper 3D structure is critical for binding catalytic activity and oral protein stability.